are waiting. Okay. I'm just going through this. Good afternoon, guys. Okay. What's the difference between Eschand and any other books? Actually, just clarify me the name of the book, Eschand. Okay, because like Eschand is a very good publisher company. It's a publishing company. Normally, we refer those kind of books in the BSc. Okay, like there are several kinds of books are there, right? But Eschand books are normally is simplified kind of the book. Okay, so normally people will refer in that time, but I'm not sure they also had a 11th and 12th book. Okay, so if it is they, just refer me, like I'll just check once. Okay, but BSc books are really good. Agarwal, VK Agarwal, someone wrote all those things. Ha, Ramesh, so you raised your hand. Okay, we can move on then. It's already 3 o'clock. Okay. I will check, Akshay. Send me the link or something. Okay. Yeah. So, I told okay, right? My Google started acting like it started like okay, Google. No, I have. Okay. Good afternoon, all. So, we are discussing about cell cycle and everything. Gopika, good afternoon. Shiran, Sushma. Okay, so what we learned in last class. So in the last class, we are actually discussing about the major. What is the purpose, right? The purpose of cell division. I don't need to repeat it, I think, right? HCOMA available in biology? No. It's not available in biology. Okay. Varma is available, but not HCOMA, I think. Okay. Hi, Harmesh. So, we are discussing about two things, differentiation. One is the cytokinesis and another one is the karyokinesis. I wrote it ulta, no issue, right? Karyokinesis means what? Division of the nucleus, and cytokinesis means what? Division of the cytoplasm, right? So these are the major things. Then we went directly went for the different terminologies, right? So terminologies, I'm repeating one more time. So we majorly have a DNA, right? What is DNA? It's a heteropolymer of the nucleotide. Good afternoon, Ashwin. Right, it's a DNA. What happens to DNA? Think about DNA as a rope. Okay, think DNA as a rope. What happens if you like start to condense it? That means start to roll it on something. Okay, so when DNA started to roll, so it becomes some thick thread-like structure, right? Good afternoon, Raksha. So we call this one as a chromatin. Okay, so it's exactly like we have a thin ropes, okay, thin lines, you can say. Okay, so from there, what's happening when you join together, you are creating a bigger kind of the rope. Okay, bigger kind of the rope. So then what happens? It's again got condensed and form a structure something like this. Okay, something like this. We call it as the chromosome, right? We call it as the chromosome. But what happens if you take a single side of this particular structure, right? A single side, then I can write something like this. So that's what we call it as the chromatid, right? Chromatid. That means what? A single chromosome have the two chromatid. Histone protein in all cells, histone proteins only present in the eukaryotic cells, not in the prokaryotes. Prokaryotes have some other kinds of a protein, but those are not histones. Okay. Good afternoon, Sanika. Yeah. So these four things should be remembered, right? DNA means a thin kind of structures, and when it's like joined together, it will form a bigger rope called chromatin. When it's further condensation, it will form a chromosome. If you take a single half of the chromosome, then you can differentiate as the chromatid, right? But what happens when this chromosome is divided into like this, right? This side is equals to this side. So because it's having the common origin, we call this one as the sister chromatids, right? Sister chromatid, because both having the similar origin. From this one only this one came, right? From that one, it's like a splitting off, right? Splitting of the chromatid. But if I take a regular body cell, then how can you find it? Like you will find chromosome like this pattern. So in this, one set of chromosome is from father or paternal origin, and another one from the maternal origin or from the mother. We call it as the homologous chromosomes, right? Homologous chromosomes. So in that, if I take two chromatids, like this one chromatid and this one chromatid, I can't call it as a sister, right? Because it's not a sister. It's not exactly alike. So I simply call it as the non-sister. Exactly. I call it as the non-sister chromatid. So simple. Okay, so think about yourself, or think about like our family system only. Okay, who we call it as a sister, right? Who we call it a sister, who actually shared a common origin, right? A common origin of our father and mother, we call it as a sister, exactly. And what we call about someone else, like those are not sisters, right? Non-sisters. So exactly like that, different origins is a non-sister, single origin is a sister. Okay, but those chromosomes will be similar in size, right? We call it as the homologous cousins. So like that. Okay, so those are the major things we discussed. So if you see this particular one video, okay, if you see this particular video, as you can see the chromosome, and they mentioned the sister chromatids also, right? So here you can see the different chromosomes are there, right? So I will write another one diagram of the this one chromosome. Okay. So here only it will come, right? Exactly. It's something like this. Exactly. So what is present on this one? As you can see, two protein structures present on this one. What are these structures? These structures are called kinetochores. Right, kinetochore. What are kinetochore then? Kinetochore is a protein which is present on the chromosome. Exactly in which point? It is present on the centromere. Present on centromere. We can also call it as the primary constriction. Okay. Constriction means what then? Constriction means like this, right? Like this. Constriction means like this. Okay, this is, I can call it as the constriction. So here is also similar. I will come back to that. Okay, exactly. So what's happening? This chromosome is made a constriction here, right? It's going in this way and again in this way. So that's why I can call it as the constriction, and it's a major constriction. That's why we call it as the primary. Okay. If there is any satellites are present, a satellite bodies we normally call it. Okay, then that constriction we normally call it as the secondary constriction. Okay. No. Because of kinetochore, no. Okay. So secondary constriction. Okay. Just I'm telling you what happens. The chromosomes will be tied together in a place. Okay. I will take a new one slide for better understanding. Okay. Give me 1 second. 2 seconds. Yeah. I will take a white screen. So that will be better. Okay. I can't see what is present in there. Okay, so think about something. Think about you are going for like collecting grass. Okay, grass are there. All these are grass plant. Okay, then you have to tie the earphone. When did I? Yeah, exactly. So I have to tie it up with a kind of thing, right? Rope. Think about these are grass, and this is the rope which I am tying this one, right? Exactly in the similar portion, how it is arranging here. Think about this is the chromosome. Okay, think about this is the chromosome. There should be something, right? There should be something tying this, right? Tying this one. So because of tying, what happened? A constriction will form here, right? This grass will constrict in a one particular region because of we are tying the rope in a very tight manner. Exactly same thing happened here. So this constriction we call it as the centromere. Okay, so centromere will be formed from first thing. Here the constriction is caused by a protein, but I'm not at all going to mention about the name of the proteins or anything, because Those things are not needed. Okay. Sometimes reading extra things is also harmful. That's why I'm leaving all those things. So it will form a constriction. About this, if you check what is happening about this, actually later the kinetochore proteins are arranging on the two sides. Okay. Kinetochore are arranging on the two sides. Kinetochore, I told you, it's a protein structure. It's not actually a single protein structure. It's formed from the multiple protein structure. Still, we are working on that. Okay, we are not sure 100% how many proteins are present in that. Okay, so we know that, okay, it's made up of a complex protein structure, almost like 49 to 54 proteins will be arranged to form a single kinetochore. Just imagine, guys. Okay, we are talking about a chromosome, and we are talking about a structure which is present on the that chromosome, and I'm telling you that particular structure have 49 to 54 proteins to form. Okay, it's so tiny, right? It's so tiny. We are actually discussing exactly. So this is how particular things will be arranged. Okay, remember about things. So if I take only this up, then it will become something, right? This is, I can call it as the chromatid, right? So this is the things. So any more doubts, guys? Because of kinetochore centromere is formed? No, because of the centromere above that kinetochore will be formed. Okay. And which protein? I told. Is kinetochore arranged outside? Exactly. Kinetochores will be arranged outside. Centromere protein, like we can leave it. Okay, no issue. Don't take attention. Exactly, it will be present outside. Can you guys tell me, like, how many kinetochore you can find on a single chromosome? How many kinetochore can you guys find in a single chromosome? Exactly, right. So one on this side and another on this side. You can call it as like one pair or two. Okay, both the things are exactly correct. Okay. How many sister chromatids are there in one chromosome? Okay, good question. How many chromatids are there in a single chromosome? You can find this one is the one chromatid and this one is the another chromatid, right? Second chromatid. So when I'm talking about sister chromatid, you can find the one pair of sister chromatids, right? Because it's not a single unit, two things are there, right? One pair of sister chromatids, you can find it in here, right? So in single chromosome, you can't find the homologous. For homologous to find, you have to have two chromosomes minimum, right? You understood? So any more doubts, guys? So question came. So if you have any question, just post it. After the question, we will solve that also. Okay. The question came. The question is simple. Mitotic spindle is mainly composed of which protein? I think you already read about the chapter I told you, right? So just concentrate. Mitotic spindle is mainly composed of which protein they are asking. Is it actin? Is it myosin? Is it tubulin? Is it myoglobin? They are asking. I'm just launching the poll. Who are in the Zoom can answer in the poll. Who are in the YouTube can answer in the YouTube live chat. Okay. What is a microtubule spindle? So simple. Mitotic spindle that we studied in the last B10 chapter, right? There will be a spindle or aster, you can say, aster, spindle, all the same, which will help in the mitosis or the meiosis process. Right. Okay. Hi, Venkat. Correct. So you have last 20 seconds. I'm not at all giving an extra time. So you have to answer quickly because the question is easy. Exactly. I'm ending the poll in last 5 seconds. Correct. So many people are giving correct answer. I'm ending the poll and I'm sharing the results. So you have to be quick, guys, if you have to answer, because I'm not giving more than 1 minute of time. Okay. So 6% chosen option A, 20% B, 64% C, and 10% D. In YouTube, everyone given option C only. Exactly. Okay. The mitotic spindle, what you may think about this, right? Mitotic spindle, you can think about the spindle. What is the other names it has? What is the other name? You can also call it as like aster in some way, right? Is there any name is there for the mitotic spindle? Aster, exactly, right. You can call it as like spindle or the aster. So from what kind of molecule it's made up of, you have to think about the structure of that, right? So it's kind of a direct question, right? So what's the correct answer? The correct answer is the tubulin. Tubulin is a protein which can form a tube-like structures, which is what makes mitotic spindle possible. Okay. But the issue is sometimes they won't give the options tubulin. Okay, then what will be the correct answer? Then another correct answer is actinomyosin. Okay, actinomyosin. So actinomyosis is like a heteropolymer of a protein. Okay, it contains a kind of actin molecules, also the myosin molecule. It's a composed molecules. Okay, not simply actin, not simply myosin. Okay, actinomyosin. Okay, so the option may be tubulin or option may be actinomyosin. Both of the option will be correct. Okay, so both option will not be given. So in between, if you get confused, like if there is no option like tubulin, only they given actin, myosin, actinomyosin, and myoglobin, then you have to go for option actinomyosin. If they given the option tubulin, you can directly go for the option tubulin. Okay, so kind of a direct question it is. So we'll move on to the next question. The shorter and longer arm of submetacentric chromosome are referred to as M arm and N arm respectively, S arm and L arm respectively, P arm and Q arm respectively, and Q arm and P arm respectively. Okay, I'm just launching the poll. I think, like, I don't know, like, how many of you remembered this one? Really? Very good. Probably you absent to that class, I think. Oh, so many people remembered the things. Exactly, right. Exactly. So many people remembered about it. That's a good thing. Huh. So they're asking like I can write a submetacentric chromosome, right? The submetacentric chromosome will be something like this. One arm will be larger and one arm will be smaller. Then we are asking what we call for a smaller arm and what we call for the larger arm. Pavan, like we discussed about the spindle in the this cell chapter, not here. Cell chapter we discussed about the things, right? When we are discussing about the centrosome, asters there. Anand, think. Exactly. So I'm just sending the poll. So I got the answers. I'm sharing the results. 9% chosen option A, 25% chosen option B, 59% chosen option C, and 9% chosen option D. So, and in YouTube also everyone chosen option C only. Exactly. Whenever we come across with a chromosome where the particular this one, the centromere is not present in the exactly half somewhere else, then we will get with two different sized arms. We normally call the smaller arm as the P arm, and we normally call larger arm as the Q arm, right? So longer will be Q and smaller will be P arm. So try to remember that. Remember guys, this is asked in NEET 2019. Okay, so I don't think that like this question will not ask. It's already asked in NEET 2019. It may be repeated in some other exams also. Okay, so you should know about these things also. Okay, good then, we will move on. So, yeah, these, why there are referred like that? Good question. So what happens when we are segregating the chromosomes? Okay, we have to know the locations, right? So later you will come to study about those things. So think about like gene is present somewhere here in this position, then how can I refer it, right? Or think about some chromosome, this part is deleted. Okay, this part is deleted due to some process. We don't know the process. Then how can I call it, right? So like calling it like chromosomes long arm, chromosome short, it's a long process. But Apart from that, we can use a different term, like one is the P and the another one is the Q. Okay, so there is no particular meaning for the P and Q. It's simply referring that one is smaller and one is larger. Okay, nothing else. There is no explanation for the P and Q. It's just referring system only. So I can simply refer it like something like gene is present in the P arm, or I can simply call the chromosome is deleted portion which is present in the Q arm. Okay, then you can directly understood. Okay. Okay, then we will come to the mitosis. There are two in P and Q for E. What should you call? Look for P1 and P2. No. Like both of the chromatids, right? Those are chromatids. That kind of structure only present in the metaphase stage. We will learn it in the next this one, next slides. Okay, where that kind of structure will be present. That's why I didn't told P1, P2 anything. Okay. So what is mitosis? What is amitosis or binary fission? Probably everyone know about the binary fission, right? So amitosis is another term for that. Okay. So binary means splitting into two, right? Splitting into two directly. And amitosis means it looks like a mitosis. I think most of you don't know about the mitosis. Some of you already know because it's completed in the syllabus, right? Sorry, CBSE. A single cell will divide into two. But this is not the mitosis. It's a different process, but it looks like a mitosis. Okay, that's what they are referring to. So we will read out what are the things. It's one of the simplest mode of the cell division. Remember, like it's like a very simple, and the one which is initiated in the it started in the initial phase. Okay, so the mitosis, meiosis, everything came later on. But what is the first thing came in the life system? For the division of the cell, it is the amitosis. Okay, so what happens? How this process will start? This simply starts with the elongation of the nucleus. Okay, before that, I have to mention where it will occur. Okay, it normally occurs in the prokaryotes. Okay, this will occur in the prokaryotes regularly in all prokaryotes. Okay, all prokaryotes where a division is happening, it will be amitosis only. Okay. But very few situation, okay, very few situation in eukaryotes, okay, in very few situation, not everywhere, okay, because eukaryotes will follow mitosis. In very few cells only follow the this kind of step, okay, amitosis step. So what happens here? Here first, the nucleus will elongate. As you can see, a cell is there. I will come to the example. Wait, okay. So what is happening here? In this particular region, the this one is there, cell is there, and this chromosomes, everything will be there. Okay, it started to pulling apart this side, both sides, right? So that's making nucleus little elongated. You can call it as the nucleus, or you can also referring this particular as the genetic material. Why I am mentioning the genetic material along with the nucleus? Because when I'm referring the prokaryotes, I have to use the term genetic material, right? Because nucleus will be absent. Karyomyosis. So it will form a dumbbell shape, that particular nucleus or genetic material form a dumbbell shape and get divided into two daughter nucleus. What happens? It will form a two daughter nucleus. Remember, the cell is single only. Okay, single cell is present, but nucleus is get divided. Later, what happens? After the nuclear division, the cytoplasm will divide and form the two daughter cells. Okay, as you can see in the last particular structure, division into two daughter cells. It's so simple, right? There is no complication at all. A cell will be there, nucleus will be there, or genetic material will be there. Genetic material or nucleus will be try to get separated. It will form a dumbbell shape. And when its nucleus is separated, then the cells will tear apart and form the two cells. Okay, direct. Remember, in this particular cell, there is no spindle formation. Okay. I will come to that. Okay. There is no spindle formation, remember, and no distinct chromosome formation occurs. This is very, very important. Here, there is not at all spindle formation is happening, majorly because it's happening in the prokaryotes. Prokaryote can't produce any spindle. But even though in a eukaryote, there is no need of the spindle because it's like directly giving a half. Okay. And here chromosome is also not forming. So you can understand, right? We are telling that DNA is there, chromatin is there, but chromosome, the condensed form, is not at all forming here. It's directly tearing apart. So here you can understand the problem. I told you, right, what happens when we try to we try to pull the earphones, which is already made knots, right? What happens? Something may be teared apart, right? One earphone may be teared apart, one half will go somewhere and another one will go to somewhere, right? Exactly. So that is what happens to the amitosis. Okay. So first thing is, this is not exactly equal. Okay, it may give rise to some equal or unequal step. And what about the cell organelles? Prokaryotes don't have the cell organelle, no issue. But it is happening in eukaryotes. The cell organelles may be unequally divided. Okay, may something contain the actual cell organelles and something may not contain the cell organelle. That's also maybe there. Okay, so it will raise to an unequal type of the daughter cells, not exactly equal. Okay, that's what we mentioned here, right? And formation of the two cells. And nuclear envelope remains intact. Remember, if you take an example of eukaryotes, the nuclear envelope, or what you can say, nuclear membrane, it's not at all going away. It's intact only, okay, without any degradation or anything. And daughter cells are approximately the two equal halves of the parent cell. Approximately. Remember, this is very important term. And these are not exactly equal, these are approximately equal. Okay, so think about a scenario where what happens during the separation of the nucleus, some part of the DNAs like get destroyed. Then what happens? Which cell will get the destroyed DNA, like that will die, okay, and only one will survive, right? Same thing happens when arrangement of the cell organelles also. If both of the cells will get the equal number of like mitochondria, then both of them will survive. If someone won't get it, then that will die. Okay. So what happens to the size? That's what we are telling. The size of the daughter cells also almost equal. We can't say it's equal. Okay, sometime it will give rise to the equal. Sometime it will not give rise to the equal cells. Okay, it's always depends upon the how the plane of division is going on. Okay, it's not predictable. Okay, like it's simply division is happening. Okay, so that's the problem with that. So if I like mitochondria divide, but it should be there, right, in a cell. It can't like start from without having anything. It should be there to go, right? Exactly. So, no, daughter cells means only two. Whenever I'm mentioning about amitosis, mitosis, it's only two. One will give rise to two. Okay. Intact means it's being there, okay, like without degenerating or without separating. Okay, it will be there only. Okay, so once you complete the mitosis, you will understand why I mentioned that one also. Okay, so if I repeat the same thing, amitosis or binary fission is the very simplest kind of the division. Okay, here what happens? Genetic material or nucleus will make a dumbbell shape and get separated. That's karyokinesis. Later, the cytoplasm will divide and give rise to two daughter cells. There is no spindle formation, no chromosome formation. Okay, there is no equal half of the daughter cells. Okay, and these are the major things related to this one. Okay, what are the problems amitosis may cause? Both of the cells will not be equal, so there may be loss of character in a DNA, right? And the survival of the two cells, two daughter cells, is also like we can't predict because both of them may survive if it's equal, or one may survive, one may die. That's also there. Okay, so normally it happens in all bacteria, all kind of prokaryotic cell. Okay, PPLO, bacteria, all those things. Okay, but if you take an example of eukaryote, someone already gave the example, right? So you can take an example of planaria kind of the organism, or you can take some of the protistans. Okay, or we can also take up the epithelial cells of the some of the amphibians, okay, some reptiles, and also the some part of the like reproductive cells of the some plants. Okay, like very like tiny few examples only. Okay, is chromosome distribute equally? There is no chromosome, but the DNA distribution is not equal. It is not equal, not always equal. Okay, it's a chance factor, always a chance factor. So it's like it's simply like taking this one lemon and throwing our knife. Okay, it may cut the knife this lemon in exactly half, or it may cut in slight other manners also, right? It's always a chance factor. We can't predict, right? Exactly like that. Not all cells, only some cells. In eukaryote, only some cells, not complete. But in prokaryotes, all cells will go through this one. Why it's called amitosis? That's what we told. This is not like mitosis. Mitosis is an organized kind of the reaction, but it's not organized one. When we learned about mitosis, you will come to know, okay, why we call it as like amitosis. Okay, this looks like a mitosis because mitosis also gives two daughter cells. It is also giving the two daughter cells, right? That's what we call it as. It looks like a mitosis, but it is not a mitosis, so it's a amitosis. Okay. Okay. So any more questions about amitosis or binary fission? So anything anything you didn't understand, or you think we should repeat some point or anything? If you don't, if you don't need anything, then we can move on. Okay. Okay, then we'll move on to the next particular one. Okay. Sir, is it unequal division? That's what we mentioned, right? Approximately 2 equal means it's almost unequal division. Okay, so that's what it's a chance factor kind of a division. Okay, it make a form what kind of division depends. Nobody knows. So we will move on to the next one. That's the cell cycle. Okay, amitosis don't have any steps. I told, right? So it's directly going and getting divided and like coming back. That's it. But when you come to the eukaryote, that won't happen like that, right? What's the driving force to divide? Bro, that increased size is the driving force. Chromatin cut like rope or condensation occurs. Is chromatin cut like rope or condensation occur? There is no condensation, Chirant, in amitosis. Amitosis, there is no condensation. If it form a condensation, it will become a chromosome, right? That's what we told. There is no chromosome means there is no condensation. Okay, the chromatins will be simply segregate or simply separate. Simple. Ropes will be there. DNA will, some part of DNA will go there, some part of DNA will go there. That's it. DNA replication, everything is done already. Okay, after only they will go for that one. So amitosis can be given to the prokaryotes, but eukaryotes are more complex. It have more organelles and more work, more kind of DNA size also, right? Amitosis is very small, exactly. Then we can't go through the exactly same like the mitosis. Okay, mitosis have to be organized one, right? What happens if you 2 or 3 people are going somewhere, you can go in like whatever fashion, right? You can go one behind another, or you can make a a row, and you can hold your hands and nobody cares because only 3 people are there, right? What happens if 30 people are going or 40 people are going? Okay, if you hold hands, if everyone hold hands and make a line, it will fill the complete road, right? Then nobody can can go in that particular road because your 30-40 people are there, right? Exactly same thing. When it's simpler in a cell like prokaryotes, it can divide simply. But when come to the eukaryotes more complex. It can't go in that particular fashion. That's why it will go through a organized way, or organized circular way, or cyclic way for the division. Okay, so that's what we call it as a cell cycle. Okay, cell cycle is a cycle, like a repeated fashion, right? So what is cell cycle then? Cell cycle is an orderly sequence of events by which a growing cell duplicates its contents and divides into a new daughter cell. What's happening? The cell is growing, the completely grown cell is getting divided and forming the two daughter cells or the new daughter cells. That particular new daughter cell again going through the same process. Okay, so when we kept on checking that particular one, we divided the cell cycle into 3 major parts. One is the interphase, second one is the mitosis or meiosis, third one is the cytokinesis. Okay, so interphase means you can simply call it as organizing phase or getting ready. Okay, so what happens in interphase? In interphase, the cell will getting ready for the next division. Okay, mitosis actually the division where division is happening. Okay, and the cytokinesis the one where actually the cytoplasm is divided. Cytoplasm is divided. Okay, so mitosis majorly contain division of nucleus, and cytokinesis will contain the division of the this one cytoplasm. Okay, so we can call it as like cytokinesis kind of sorry, it's cytokinesis, and this one is the karyokinesis kind of a thing. Okay, so when you group together all these, you can call it as the cell cycle. Like get set go. Exactly. After I interface, it's like get set go. Okay, so you can see the cycle here, right? So exactly same thing will work. Your body cells will be in this particular one. Okay, we call it as the G1 phase. From there, what happens? It will go through a particular kind of a nature. We call it as the interphase, all this. And when it is completed this interphase, it will be ready for dividing. It will complete the division. Okay, so G1 plus S plus G2 exactly will become, right? So interphase is the longest phase in your cell cycle. Okay, and in mitotic phase is the comparatively smaller, and cytokinesis is very tiny, a fraction of time it will take. Okay, interphase is also called as resting phase. Exactly, we will come to that once again. Okay, so this is a complete cycle. Once it completed the cytokinesis, what it will do? Once it will completed the cytokinesis, it will again go for interphase, again do this. Okay, it's a cyclic. It's doing the same thing again. Okay, without any like tension or anything. Okay, it's again and again going because it have to give the new cells, right? So that's what we study. But cell cycle, remember the steps will be interphase, mitotic phase, okay, mitosis, or we call it as the, once again, we also call this one as the mitotic phase, okay, mitotic phase you can say, and the cytokinesis. Okay, so the time required to complete one cell cycle is called the generation time. Okay, so why we call it in that way? Because it will give rise to a new generation, right? We normally say, right, if you are like grandfather or grandmother says something, then or your father or mother says something to you, like your hairstyle or what's your makeup or dressing sense, so then you will say like generation gap, like you won't understand, it's the generation gap, right? So saying what is the generation then? Okay, generation means giving from one particular area to the next one, right? So think about your father, everyone will be there, father, mother, they will be living in a particular kind of era, and next you will become like you, right? Next your daughter or son, your progeny, right? We can say daughter and son. So that's what we call it as. This one is one generation, this one is one, and this one is versus another generation, right? Exactly. The period required to complete one cell cycle is the generation time. One cell will be there, okay? So it will go through a cell cycle and it will produce the two cells, right? So this particular time you can call it as the generation time. So exactly, Jeevan, the generation time is for a single cell, okay, not about a complete organism. It's for the single cell. So generation time for the complete organism is different, right? Generation time of the humans may be like some years, right? Like that. But what we are referring here is for the only to the particular cell, not for the organism. Okay, so example, generation time always varies. Okay, generation time is differ from different organisms. If you take a yeast, it will be 90 minutes. Okay, 90 minutes, what happens? A generation time will be over and it will produce the new cell. But if you take a plant's root tip cell, what we normally study in our labs, that's why they gave that particular example, it will take a 20 hours. Okay, almost like a day, right? But if you take a human cell, it's exactly 24 hours. Okay, don't think about minutes, second, everything. It's always an approximation. Okay, so it may take like 23 hours some minutes or 24 minutes hours some minutes, like that. So approximately 24 hours it will take. So what you can find out here, look, the yeast is a simpler organism, right? So what's happening? The generation time is low, like it can divide quickly, right? But our human cells are more complex. so it is taking more time, right? So as the complexity of the cells increase, the division time also increase. Okay, so I think you understand that one, right? The phases are interphase, mitotic phase, and cytokinesis. Once it's time to complete this particular stage or complete cell cycle is called generation time, and the generation time will vary from organism to the organism, or I can say simply say it like cell to cell. Okay, like some cells, if you take a stem cells, meristem cells, those are very quick. Okay, like quickly they will divide because a lot of things it have to do, right? But if you take a regular like stagnant cell, it will take a lot more time. Okay, so that's related to the phases. So we came to this. So then we will try to learn about the phases in a more detail. Okay, the first phases is the interphase. Okay, so interphase is the major phase, and this phase will take more time or more time in the complete cell cycle. Okay, so how it will be visible under the microscope? Okay, if you like take any cell in your body, and if you observe, if it is in the interface, it looks something like this. Okay, everything like no change. The cells will be there, and nucleus will be there, and DNA will be like spreaded like the chromatin. Okay, there is nothing will be there. Okay, so you can't differentiate any cell cycle or anything. That's why in the initial days we called it as the resting phase. Okay, we called it as the resting phase. What we thought is like, okay, cell will undergo a division, then it will become completely like what we can say, it will out of energy and everything, so it is taking a rest there. After taking a sufficient amount of rest, then it will again come back to the division, we thought. Okay, because regular compound microscope we are watching. Okay, when we discovered the different kinds of the molecule interacting and new kinds of the techniques come to our like to child to adult, we thought like that. Okay, but when the new techniques came, then we come to know that the resting phase is not actually a resting phase, because this particular interface is the one of the most how can I say engaged kind of the phase. Okay, most engaged phase, most active phase in the cell cycle. Okay, so we thought it's a resting phase in initial days. Okay, only in the initial days. We normally say, right, what you see probably a lie, nodiddu sullagabodu, right, like that. So exactly same thing. What we first seen, we thought that okay, it's resting, it's a lazy thing. Okay, it's a lazy kind of the phase. Okay, then we come to know that no, it's not lazy, it's actually super active, but we can't see through a naked eye, because everything what is happening is a chemical reaction. Okay, all the chemical reaction is happening. Okay, they thought like that. So that's what made us curious regarding the interface. So we studied in detail. So after studying in detail, we came to know that okay, interface can be divided into three phases. Okay, based on the different checkpoints, like different stages. Okay, so we call it as G1 phase, S phase, and G2 phase. Every phase have its own value. Okay, every phase have its own value and it have its own function. Only when it completes its function, then only it will move on to the next phase. Okay, think about this is like a factory and it's manufacturing something. Okay, so think about what it's manufacturing. We will take a simpler thing. Give me some example. Okay, what you want to manufacture. First example, I will take that as an example. So I don't think about any example. I'm not at all thinking any. Pen, exactly. Okay. Ananya. So think about a pen. Okay. So we are running a factory which is having a pen factory. Okay. We have a pen like this and it's have a nib and everything. Okay. So what is the first thing we can prepare? Okay, we have to prepare a model of the pen, right? Because to make hundreds of thousands of pen, we have to make a model of this particular pen, right? So we will create a model in this particular one. Okay, so without creating a model, you can't go through the next phase, right? So next phase is the creation phase, creation of the pen, right? So what happens? Model creation will happen in G1 phase. What happens in S phase? Synthesis. That means production will happen. Model is ready. Then what will happen? the production phase will happen in the S phase. Okay. So after production is what? You will directly give it to everyone. Nah? No. If it is something wrong, then blame is on your factory, right? They will say like, no, this pen is not good. Sometime I took like giving 10 rupees, but it's not at all working now, like they will say, right? That's why in the G2 phase, what happens? The final steps will happen. Final steps almost will be like checking exactly and refilling exactly. Okay, refilling, checking, everything will happen in the G2 phase. Okay, so what happened then in G1 phase? It made ready. Okay, so our factory is ready, our model is ready, our worker is ready. Okay, everything is ready. Okay, getting ready. It's almost like getting ready. So then what happens in the S phase? It's actually doing the process. Okay, production is happening, pen is happening, worker is doing, pen is done, everything. In G2 phase, it's checking like is pen is working properly and it's refilled properly. Okay, you still need anything, right? So all the things will happen. So then it can go for the writing and all the process, right? So like that, it will divide into 3 phases. So that's what we did. Okay, G1, S, and G2. We will add. So no, like we won't need ad. Okay, ad will give the, so ad will take most of our money. So we don't need ad. We need customer review only. Okay, so we will move on. Okay, so if we keep this particular one, so we will study in detail about G1, S, and G2 phase. Okay, the first one is the G1 phase, or we normally call it as the gap 1 phase. That means what? That means this is the first gap what we are experiencing. Okay, so why we call it as like gap 1 phase? If you think about this particular, I'm coming back to this particular cycle only. Here you can see after a particular kind of a division, okay, a complete mitotic division, the cell is coming into the rest, right? This is the first rest or first gap. That's why we call it as the gap 1 or rest 1 phase. Okay, rest 1 or gap 1 kind of the phase. Okay, so what happens here? The cell growth occurs in this phase. That means cell is already completed its division. That means it's a newborn. Okay, it's a newborn kid like. So it's getting ready. It's growing. Okay. Then what happens? The structural and functional proteins are formed. Okay, the proteins which are present, right? So there will be like hundreds of proteins. We need thousands of proteins. All the different proteins will be formed. And another term I have to add here, organelles will be arranged. Organelles will form and arrange. What happens when it is going through the cell cycle step? Okay, when it's going through a cell cycle step, it's actually eliminated all of its organelles. Why it did that? Because if most of the organelles are there, then division of DNA is almost like problematic. Okay, so somebody will stuck there, some organelle will stuck somewhere here, and it can destroy the DNA. Everything will happen, right? So it almost removed every organelle. But when it's come back to the G1 phase, it will produce all the organelles and it will produce all the proteins and everything. Okay. And nucleolus, what it will do? The nucleolus will produce all the different kinds of RNA. So why we need RNA? So RNA is needed for the production of the protein, right? rRNA will produce the ribosome and mRNA will produce, bring the, one second. mRNA will bring the particular kind of the message, and the tRNA will help in the this one production of the particular kind of the protein, right? So nucleolus will produce the different kinds of mRNA, so RNA molecules. Okay, and metabolic rate of the cell becomes very high. What is the metabolic rate then? Okay, metabolic rate means how much energy it can expand. Okay, energy expenditure, we can say energy expenditure. You just take an example. Okay, we will take 2 people as an example who need most energy. One person who is doing work, okay, who is doing the agriculture work. Another person who is lying on his bed throughout the day, right? Throughout the day, who needs most energy? Exactly. The person who is going for the agriculture work is need more energy, right? Because it's doing a lot of work. Exactly. Here, nucleolus production is happening, RNA production is happening, organelle production is happening, everything is running. So it needs lot of energy. It will consume most of the energy molecule, and it will grow big and it will expand it. Okay. And so that's why it's growing. The cell is, size is becoming big, big, big. Okay. It's increasing. Okay. Energy requirement is more. Exactly. As you can see in GIF, like it's slowly increasing. The cell is increasing, right? Exactly. So it may be also called as a pre-DNA synthesis phase, because here DNA synthesis is not taking place, but what is that? That is simply a cell is like increasing its side and like exploding something. Okay, just thought of adding it. Huh. So here DNA synthesis is not happening, but what is happening? All the other molecule is happening. Okay, so all the other molecule which is required for the cell cycle is production is happening. Okay, and remember, in this particular step only, all energy molecule, that all requirement will be filled. All requirements will be filled for cell cycle. Cell cycle is an energy needed structure. Okay, so for a cell to get divided, it needs a lot of energy. Where this energy molecule, all the protein, carbohydrate, everything is accumulation is happening, that will happen in the G1 phase. So simply I can simply call it as the growth phase, right? A growth phase, because everything is happening here. And we also can call this one as the post-mitotic phase. Why we can call it as the post-mitotic phase? Can anybody tell me? Like this question is for you guys. Why we can call this particular phase as the post-mitotic phase? So is there any reason behind that? No, like exactly. Okay, this is happening when? Okay, it's happening after the mitosis, right? Once mitosis done, like I will take an example here. One cell went through mitosis, give rise to the two daughter cell. This daughter cell is going through this G1 phase, right? That means what? So after the mitosis, when a new cell is formed, a particular new cell is going through this process. So that's why we call it as the post-mitotic phase. Okay, post-mitotic. So after the mitotic, this particular phase can be found. So what's the important thing here you have to remember? G1 phase is called gap 1 because the first gap phase it is, and it can be also called as post-mitotic because it will occur after the mitosis, or you can also call it as the pre-DNA synthesis phase because after this phase, DNA synthesis will taking care. Here, most of the organelles will be arranged or produced, and all the energy molecule or biomolecule production will be happening. So it needs a lot of energy, so metabolic rate will be high. Okay. Exactly, Venkat. So any doubt regarding this particular phase? Any doubt regarding this particular phase? If you have any doubt, just put it in the chat box. Otherwise, click on yes, so I can move on. Okay. So if you have any doubt, yeah, exactly. Then we can move on. Okay, nobody have doubt. So second one is the synthesis phase. Okay, so as the S suggests, S means synthesis. Synthesis means something is production is happening, right? So what will happen here then? In this particular phase, the major thing is replication of DNA takes place. Okay, what's the meaning of replication then? That's the question, right? Replication means the DNA like this, right? The DNA like this. What happens? This DNA utilizes some of the enzymes, okay, not condensation, I will tell, okay, it will utilize some of the enzymes. What it will do, it will create a exactly same copy of this one, okay, exactly its own copies, okay, exactly its own copies. So how can it able to create like that, right? How can it create something which is exactly like that? So it's majorly with the our nucleotides, because you know that A always pairs with T and G always pairs with a C, right? What DNA will do? DNA have the two strands, right? First, it will separate its strands, okay? It will separate its strands something like this, okay? It will separate its strands. Then, like matching is easy, right? So if it is A is here, then we have to join the T. If G is here, and we have to join the C. Same thing, right? Exactly. If A is here means T will be here, then we have to join the A. Understood? What happens? The DNA is made up of two strands. You know that DNA will separate the strands. Okay. After separating the strand, the simple thing is we know that nucleotide always pairs with the purine with the pyrimidine only, and A always with T, G always with the C. So it will just make a matching. Okay. After making a match, it will get the two strands. Simple. Okay. So this color strand, white color strand came from what? Strands is made up of the nucleotides, right? This is only Ananya. Okay, this strand is made up of the nucleotides. I can write something like this also, right? So these nucleotides are formed in the cytoplasm. So enzymes will help in joining those nucleotides and forming a strand. Okay, enzymes will do that particular work. Okay, so that the strands are coming from there. So this is what called replication. Okay, this is what called as replication of DNA. So this is what happens, replication of DNA. This is what happens in the S phase or the synthesis phase. That means DNA get doubled. Okay, DNA will getting doubled. That means double ho gaya. Okay, then protein molecule called histones are synthesized that cover each strand of DNA. Okay, we created the DNA, but what we need next? We need histones. Why we need histones? We need histones because that is what create the DNA molecule arranging fashion, right? So not fan, some work is going on. Okay. Yeah. So what happens? This histone proteins are needed for that particular DNA to arrange on itself, right? So histones are also produced in this particular phase only. Okay. DNA is also production is happening, and histone is also production happening. Okay. So these are the major two things. We will come to the third point later on. Okay. So what is the other thing then? One second. Chat is not responding. Okay, so as you can see here, so we will take an example of something like this. Okay. Like I think you guys can hear me now, or like give me 1 second, guys. Like it's stuck working. Okay. So can you guys hear me? Hi, aeroplane starting. No, not aeroplane. I will keep it as something like this. Okay. So how can I come back to this? So DNA will be there in the initial phase, in the G1 phase. So G1 phase, the chromatin will be something like this. Okay, chromatin in G1. Chromatin in G1. So what happens in G1 after the G1? Okay, this chromatin is getting okay. 1 second. So think about this. It will be a single chromatin thread, but what happens? This single chromatin thread, due to the replication, it's dividing into two threads, right? One thread is forming the two threads. I can write the thread something like this. Okay, so chromatin in G1, chromatin in S, chromatin in this one. Okay, two DNA is formed in this particular one, right? We can call it in that manner. 1 second, guys. So everything is it literally stuck here. That's why I'm like closing everything. Then I will come back. Okay. Zoom is not working. Huh. Oh, why it happened now? Yeah. So I think everything's sorted now. Who are in YouTube, also wait for like 1 or 2 minutes. Okay. I'm just trying, but nothing is working. Chat is not responding and Zoom is not responding. Okay, we'll come back. Yeah. So what happened here? So instead of having a single chromatin after the a particular kind of S phase, so it's formed two kinds of the structure or two chromatin. Okay. In a simplified manner, how can I write it? This one. Okay. So I can write this kind of the chromatin which is present in G1 will form something like this. Okay. This one in the S phase. So what is happening here? What is happening is the DNA duplication is happening. Okay. DNA duplication is happening in this one. Okay, I'm not sure about the chart sections, guys. Chart section is not working, so I will wait for some other time for the chart to work. Okay, I think you guys can hear me, right? That's the thing. Yeah. So G1 and S phase, how the differentiation happening? DNA doubling is happening. Okay, DNA is getting doubled. A single thread is forming two threads. So this is where our sister chromatids are forming. Okay, but still you can't find it because condensation is not happening, only replication is happening. Okay, sister chromatids are forming in this synthesis. Hello, can you guys hear me? Yeah, I think you guys can hear me, right? Yeah. We'll move on then. Where we are somewhere in between the G1 phase, sorry, S phase. Okay, so this is what happened in a space. Replication will happen and protein molecule histone production will happen. Okay, So in that phase, another one important thing is happening, that is the centrioles, if it is present. Okay, centriole will replicate in latest phase. So what's the thing about this then? The centriole will be present a single organelle in each cell. Okay, centriole will be present as a single organelle in each cell. Okay, this single centriole will replicate to form two centriole, because we need two centriole in two poles to pull the chromosomes, right? So that is what. So is it chromatin replication into chromosomes? No, Venkat, remember the difference between the chromatin and chromosome is, one second, the difference between chromatin and the chromosome is the how the condensation is there, right? How the condensation is there, nothing else. Okay, so chromatin replication is happening, that almost is equal to the chromatid replication. Okay, so condensation happens, this will looks like, otherwise it will looks like this. Okay, resume recording, I don't know, like my laptop is working like a weirdo. Okay, it's going to the first particular one. Yes. So late phase means in the end, exactly. Late phase, centrioles replicate in the late S phase means in the end of the S phase. Okay, in the end of the S phase. So any questions regarding S phase, guys? Any question regarding the S phase? Sorry, some problems is happening. So sorry for that. Okay, any question relating to the S phase? Like, okay. So we will join once again off my video. Okay, what happened? So third one is the G2 or gap 2 phase. Okay, probably you now understand why it's called gap 2 phase, because it's not a gap 1 phase, right? So gap 1 phase is there, and this is the second gap phase, right? So that's what the important thing. So after the S phase, it's coming here. So in this, what happens? Tubulin protein synthesis starts for the spindle formation. Okay, tubulin protein, which is present in the spindle, right? So we are coming back to this once again. So that particular tubulin protein production will happen in the G2 phase. Okay, because it have to produce the spindle fibers in the next cycles. That's why. And this phase you can also call it as post DNA synthesis phase, because already DNA synthesis happened, and this phase is after this DNA synthesis phase, right? So you can call it as the post DNA synthesis. Okay, so cell division involves enormous expenditure of energy. So cell stores ATP in G2 phase. So cell division will need a lot of energy. So what it is doing, the G2 phase is creating a lot of ATP synthesis. Okay, what you can see here, right? So some machinery, something is there, a machine working, a kind of things is happening, right? So this is how actually ATP synthesis will happen. Okay, all these are different kinds of protein which will be present on the membranes of the mitochondria and the chloroplast. When different molecule pass from here to here, especially hydrogen molecule or protons, it will utilize that kinetic energy and convert the ADP molecule, adenosine diphosphate, into the ATP molecule. Okay, so I just wanted to put this one, mitochondrial ATP synthesis production. Okay, so ATP needs a lot of ATPs are needed, so everything is created in this particular phase. Okay. Next, after G2 phase, cell enters into the division or M phase. So G2 phase is the last phase of the interphase. Once it's done, okay, it's ready for the cell division. It have the energy molecule, it have the spindle fibers, it have the proteins, and DNA is already replicated. Everything is ready. So it's next ready for the division. Okay, G2 means very simple. Okay, so any question related to the G2 phase or any doubt regarding G2 phase? So I'm just waiting, guys. Any doubts? Otherwise, click on yes. So like I will get to know, right? Okay. What others are doing? Like, huh? Okay, then if you have any doubt, just put it in the chat box. Okay. So, huh? Next one. Not all the things will go in this manner only. We told, right? Some third point once again. Okay. Third point. So cell division needs lot of energy. So for that particular energy, we need ATP. ATP is the energy molecule of the cell. So production of ATP and storing of ATP will happen in the G2 phase. Okay, cell division need a lot of energy, right? That energy is the ATP only. So an energy molecule. That energy molecule production will happen in the G2 or gap phase. Okay. So where will be the ATP will be stored? ATP will be stored in ATP form only. ATP is a free molecule. Okay, it's not stored in a structure or anything. Storing of ATP means production and keeping it in the cytoplasm. Okay, ATP is a free molecule. It will be directly will be there. It's not stored in any structure or anything. Okay. So not all the things will go in this particular manner only. For going in this particular manner, it's going for the this one, cell division. But some of them don't want cell division. We already seen that, right? Those are like completely permanent kind of cell, we call it, right? Those will enter a particular cell stage, we call it as the G0 stage or quiescent phase. Okay, quiescent means without any changing. Okay, it's keeping in the same way. So what happens in this one? The cells which are not to divide further do not proceed beyond the G1 phase and start undergoing differentiation into specific types of cells. Such type of cells are to be called as quiescent cells or G0 phase. Okay. What happens? Some cells after getting divided, so it will be in the G1 phase. In the G1 phase, so they don't want to go for cell division. Okay, they want to change their morphology. Then what it will go? They will start to differentiate. We will take an example of neurons. Okay, what happens? Neuron cells will get differentiated. So then it can't go for any kind of cell division. So we call that particular kind of cells will be in the G0 or in the quiescent phase. That means they never they will not go to the cell division cycle. Those are like a stable kinds of the stage. Okay. These types of cells do not divide after attaining full differentiation. Okay. They will not going to divide or doing anything. Once it's finally differentiated, it will work until it will die. Okay, so then it will die after eventually, right? So example, you can take nerve cells or neurons or skeletal muscle cells or mature circulating white blood cells or the red blood cells. We will take an example of RBC. Okay, RBC is a very good example for this. Why? Because when RBC is forming, it will be having a nucleus, mitochondria, and all the organelle. Okay, I can write it as the immature RBC. Okay, it's almost circular. Okay, a round kind of cell as the regular cell. Okay, but when it's going through a maturation or differentiation, what it will do? It will lose its nucleus. It will lose its cell organelles. Okay, all the organelles it will lose. Then it will become a mature RBC. Mature RBC will be something like this. As you can see in the structure GIF above, right? Mature RBC. Getting maturity now, or achieve maturity. We can say that. As you can see in the GIF, right? It will forming a concave kind of the shape, right? A concave shape. When it will attain that particular concave shape, when it's losing the nucleus and all the organelle, why should it lose it then, right? Why it's losing the nucleus? Because nucleus is the center, everything. It should lose the nucleus because nucleus and other organelles need a lot of energy, but the function of the RBC is just to carry the oxygen, right? And it have to accumulate more hemoglobin also. That's why it will normally lose all the other things. Only the cell membrane with the hemoglobins will be left out. Okay. So inside that, what will be there? Inside only the cytosol will be there, nothing else. Okay. The cytosol will be there, no other organelles will be there. Okay. So that is what happens to the other kinds. But not always nucleus will be given or withdrawn. It's only happening in RBC. If you take an example of neuron, so what happens in a neuron cells? A circular type of cell will differentiate and form into a different kind of the dendronic cell or neuron cell, right? Here it's changed its morphology. That's why it can't get divided. So we call it as the G0 or quiescent phase. Okay. So any doubt regarding G0 and quiescent phase? There are types of G0 and quiescents are there. Okay. But that's not in the syllabus and never asked. So I'm not going in depth of that. Okay. In go phase, it is the condition which all the examples like RBC, sir. In go, I didn't understand, bro. Is it the condition with all the examples like RBC? No, only certain type of cells only go for G0. Others never go to G0. Okay. Lifespan of the neurons will normally don't die, or any other cells in our body. It's always dependent upon its particular work. Okay, if you take an example of a RBC will divide die soon, like within 3 months it will divide. If you take an epithelial cell, it will take some other time. It always differentiate, even neurons also. We can't I simply can't say that. Okay, so it depends. RBC then immature stage, or it's formed in a mature stage. Hemoglobin is a protein, so it will be formed in the initial stages of its development only. Okay, hemoglobin will be there before it's becoming mature. Okay, like hemoglobin production will be happening, so that will be arrangement is happening. Once everything is done, nucleus, everything will be removed off. There is an example of RBC. Is it same for nerve cells? Now cells. Differentiation means the differentiation of the this one structure, right? RBC differentiation means removing of the nucleus. So everywhere differentiation is there, but type of differentiation is different, right? By losing, isn't it become less stable? So that's why it's dying little quickly. Okay, it don't stability means it don't have the work, right? Apart from carrying the oxygen and carbon dioxide, it don't have any other work. So it doesn't matter to that. Okay, so our body is keeping in that way. Okay, any more questions, guys? So okay, then I will complete this one and we can close this after this. Okay, so I told you like G1 phase, it will go to S2, it will go to G2, then it will go to mitotic. Like who will be controlling that, right? Like who will ensure that all the proteins, everything produced in G1, all the DNA is exactly replicated in S, and ATP is produced properly in S, G2, right? There should be someone, right, who is taking care of all these matters, right? So those kind of things we call it as the checkpoints. Okay, what are checkpoints then? Checkpoints are the one exactly like our checkpoints which will control the cell cycle. Okay, so majorly we divide the checkpoints into 3 different checkpoints. Okay, where actually everything will happen. The first one is the cell growth checkpoint, or we can also call it as the G1 checkpoint. Okay, what it will do, it will occur in the end of G1 phase. Okay, so what it will do, it will check that is cell is big enough for the division, okay, or it have made up of all the proteins or synthetic molecules, enzymes. Okay, everything it will check. Think about a cell, it just not produced a particular kind of a protein, but it's required. Okay, cell cycle. Then what it will do, it will say, okay, you are not qualified, you are not produced the protein, go back to the G1. Okay, it will again send that to the G1. Once all the requirement match, it will not allow this G1 checkpoint, not allow the cell in the G1 to enter to the S phase, because it's a disaster. Okay, if it has entered to the disaster, if it don't have the protein, then it's completely wasted, right? The cell will be getting to it will die, right? So that's why. Okay, some cells passed the G1 phase, then it went to the S phase. Okay, so S phase, what will happen? DNA synthesis will happen, right? And after the end of the S phase, you can also find another one checkpoint. That's what we call it as DNA synthesis checkpoint or S phase checkpoint. Okay, so what happens? It will do that the DNA replication happened correctly or not, because during the DNA replication, a mutation may be happened, or a chromosome may be missing, or it's not at all did the this one synthesis or anything, right? So what it will do, it will check that, okay, replication is done and the replication is correct. It's already checked, verified, everything. Okay, if it is verified, then it will say, okay, you are allowed to enter to the next stage. If it is not, then it will say, okay, then you again go for the DNA synthesis, do all the things and come back. Okay, So if some cell pass the S phase, then it will go to the G2 phase. But remember, G2 phase do not have any checkpoint. Okay. So the question once asked, right? After S phase, where you can, it will be controlled. Once a cell is past the S checkpoint or DNA synthesis checkpoint, there is no checkpoint in G2. Another checkpoint will be present in the M phase. Okay, will be present in the M phase. That's called as mitosis checkpoint. So what happens here? In this particular checkpoint, the cell will only check mitosis complete or not. Okay, mitosis complete means karyokinesis, cytokinesis should be completed, right? So, sorry, karyokinesis should be completed, right? It will check that, okay, DNA separation happened, karyokinesis is done, and everything. If it is not done, then it will say, okay, just wait for the cytokinesis. Okay, just wait. Yeah, everything is not done yet, so you have to wait. If karyokinesis is done, then it will send a message that, okay, our end is done, karyokinesis is done. You can go for the cytokinesis kind of the procedure. Okay, so it's repeated. I'm repeating one more time. Okay, checkpoints are the particular points in the cell cycle where the cell can regulate what is happening with it. Okay, so we will normally find three different checkpoints, and one in the G1, another one in S, another one in the M phase. Okay, the first checkpoint is cell growth checkpoint, which occurs in the G1. It will check production of protein and all the things. Okay, if everything done, it will allow that particular cell to go to S phase. The S phase checkpoint will check that DNA replication happened properly and everything, and histone protein. If everything is done properly, it will allow the cell to go for the G2 phase. G2 phase, we can't find any checkpoint. The next checkpoint only present in the M phase. It will only check that the karyokinesis is done properly or not. Once it is done properly, once karyokinesis is done properly, then this checkpoint will allow the cell to go for cytokinesis or division of the cells. Okay, then again the same thing will repeat. So these are related to the checkpoints in the cell cycle. Any doubts regarding cell checkpoints, guys? Any doubts regarding checkpoints? I just launched the NPS. Okay, if you have any doubt, you can ask me. Otherwise, how do you know all days? What days? Didn't understand, Pawan. So if you, how do we know all this? We know all this because we interfered in the cell cycle. Okay, what we did is like we deleted some of the proteins from the G1. Okay, so then we will see it is going through S phase or not. It is not at all going the S phase. Then we conclude that okay, there are some pointers are there. Then in the new biochemistry, when we found out and what we are discussing, then we come to know that what proteins are doing this and everything. We call it as CDK. Okay, CDK cell division kind of the thing. So those will be controlling this kind of the structures. Okay, some proteins will be there. Then we will see that okay, this one is controlling this one. This protein is controlling this one, like that. If there is, exactly, good question, Sachin. Sachin is asking, what happens if something is wrong, if it is allowed? Then the cell will divide or cell may turn into a cancerous cell, right? So there may be a mutation happened or cells can't divide if there is no enough energy or anything, or if DNA replication is not proper, it may turn into a cancer cell, right? All of that are problematic to the body. That's why it should follow that. After many days listening to what cell is most protected? All cells are like all cells are protected in this way only. Okay, but I didn't understand. You are meaning also same or different? In YouTube, any doubts, guys? If you have any doubts, just put. Otherwise, we can close the class. Undercover agent. So humans and further organisms comes under the mammalian class. Can we think that humans and blue whales are same ancestor? Exactly, Varun. We are from the same ancestor. All mammals from the same ancestor only, but later it's divided.